recording. Great. So thanks everyone for joining us today for our Adaptation for Communities lunchtime learning sessions. We record these sessions and upload them later on to our YouTube channel and website. So just again, if you prefer not to be recorded, you can please use the chat to ask questions or turn your camera off. So our Adaptation for Communities lunchtime learning sessions are part of an ongoing climate leadership program as well. It's kind of designed to share practical skills, leadership skills, and place-based learning and inspiration. So today we're joined by Kerry Dodd from Buglife, exploring solutions and resources in Forth Valley kind of setting and nationally. We're trying to understand how climate adaptation can work in our own communities and spaces. So I'm Steffi, I'm joined by Isabella from FEL Scotland. So if you need anything, we're the ones chairing the session today. And this is just part of our broader work as Forth Valley's Climate Action Hub. So through these series, we're aiming to raise awareness of achievable climate actions in our region and shine a light on some of the incredible work that's already happening. Today's focus is nature at risk and a sort of dip into citizen science. So we hope that you really get some good information from our lovely speaker, and feel free to ask any questions or discussions at the end. But I will leave that for now and move on to our speaker. So thank you, Kerry, if you're happy to take us away.
Yeah, I'll just share my screen. So, hi everybody, I am Kerry Dodd, and I am one of the Guardians of Our Rivers Conservation Officers at Buglife. So I'm going to go through about Guardians of Our Rivers. Once I've done that, I'll talk about climate change, and then I can talk about how you can get involved in Guardians of Our Rivers. Oops, slides being a bit slow. So Guardians of Rivers, so we are the Riverfly Hub for the Hub for Riverfly Partnership in Scotland. We deliver training and support to community groups so they can monitor the health of their river by surveying the invertebrates. We're in the second phase of this project, which is lottery funded, National Lottery Heritage funded. So we were able to have a part-time officer join the project. We are bringing on new groups to train in this phase of the project, but we're continuing to support our existing groups. We're providing them with refresher training, upskilling them into an advanced course, and then we're also looking to train a network of volunteer trainers to make this sustainable. So there are three main reasons that people get involved in Guardians of Our Rivers. The first is restoration. So here we've got a picture of the River Mick, the Spittal of Mick. And here the Balmoral Rangers, the Cairngorms Volunteer Rangers, and the River Dee Trust have worked together to plant some trees. You can see those little pegs on the side of the bank there. So that will provide much needed shade. And you can see there in the middle some large woody structures. So these are wind-blown trees that have been repurposed and put into the river to create habitat. That central photo is representing pollution. So this photo we can see some sewage fungus just downstream of an outfall pipe, and pollution and water quality are probably the main reasons why many people are getting involved with Guardians of Our Rivers. It's in the news a lot. People are reconnecting with their water courses. I know that for myself, for me, I really reconnected with my local burns when during lockdown walks. I went and walked along a couple of burn that I didn't really even know existed. It took me around some places that I've discovered, and I think that's the same for a lot of people reconnecting with nature after lockdown. And then we have community. This is a group here that we had two groups. So we had this was a training session on the Kilmartin Burn just outside of Loch Ilphead. We had a group from the Loch Tarbert Action Group and a group from the Heart of Argyll Wildlife Trust. And they came together to do their training. And it was brilliant because the groups were aware of each other, but they hadn't really formally connected. So having their training session in the same location meant that these two groups were able to make that connection, and now they can share resources, share ideas with Riverfly, but also outside Riverfly. So why use invertebrates? Freshwater invertebrates play a vital role in maintaining clean water. They help break down and filter organic matter, and they provide a food source for fish, birds, and mammals. And their presence is an indicator of the health of freshwater habitats. And this is because they're sensitive to changes in water quality, changes in habitat diversity, and changes in water level and flow. So at Guardians, we're training the groups in what is known as the Riverfly Monitoring Initiative. And this is a citizen science survey to monitor water quality of rivers and to detect any decline in that water quality. The survey is based on a low-level identification of 8 invertebrate groups, and there's a simple scoring methodology. And the simplicity ensures a robust and reliable method that can be taught in workshops and consistently applied in the field by trained volunteers. The 8 groups have been chosen as they're sensitive to pollution, and they stay in the same location for around all year round broadly. So any decline in their numbers suggests that there's a problem with the water quality rather than due to their migration. So we at Guardians, we deliver the training in 2 parts. We have a theory session, which we deliver over Zoom, and a practical session in person at a chosen monitoring site. And both of these training sessions last about 3 hours. And once the training is complete, the Riverfly groups survey their site or sites monthly from April until October. And the sampling uses what's called a 3-minute kick sample, followed by a 1-minute hand search. And this is the same method and the same equipment used by statutory authorities such as SEPA, the Scottish Environmental Protection Agency, but also ecologists, water companies, universities. Everybody's using the same standardized method, which is really important. So with each kick sample, once we've taken the sample, we turn the sample out into some trays. You can see in the photo there, and we look for the 8 target groups, which I'll show you in a minute. We then use a simple scoring method. So the higher the abundance, the higher the score. And this score for each of these taxa are recorded on a database called Cartographer, which is a citizen science open access database. So if you're part of the citizen science project, you may already have access to Cartographer. The great thing about it being open source means that anybody can access it. So there's people from SEPA, Scottish Water, there's universities, there's students, schools, and just other riverfly groups all looking at this data. So the 3-minute kick sample, you can see here, this is me taking a kick sample. I've got my buoyancy aid, a long pair of gloves. So when I'm doing that 1-minute hand search at the end, I stand with my net downstream and kick in the water, disturbing the substrate so that the substrate loosens and the flow of the water carries the substrate and the invertebrates into the net. 3 minutes isn't just standing in one place, it's zigzagging our way upstream, intercepting different habitats because different invertebrates live in different habitats. We want to sample the slow flowing, the fast flowing areas, areas where there's silt and sand, areas where there's gravel. So we need to intercept all of that, also submerged vegetation. And the timing is the timer is only going when you're actively kicking. So while walking in between those different sites, the timer is not going. So a 3-minute kick sample will take much more than 3 minutes. And then once we've done that, we go over the same stretch of river and we do what's called a 1-minute hand search, picking up some large rocks, giving them a rinse off in the net. We have some invertebrates, especially the ones known as stone clingers, that like to cling to stones. It's a little bit like when you pick up a rock in the park or in your garden, find all those things like woodlouse underneath it. So we also lift up some of the larger boulders and just allow the river to let those that are clinging to the underneath of the rock to flow into the water. And then that's how we take our sample. And once you've tipped it into our trays, we are looking for our 8 target groups. So this is a picture of the guide that our volunteers are given once they've done their training. And these show the 8 groups that they're looking for. So first of all, we have the caddisflies. Caddisflies, their name order is Tychoptera, and that's named for the adults. The adults of caddisflies are slightly brownish. They look a little bit similar to moths, but moths have scaly wings, whereas caddisflies have hairy wings. And caddisflies get split up into two different groups. We've got the cased caddis here. Cased caddis spin a silken thread, and they use this thread to bind material from the bottom of the riverbed, to create a case. So they can use small stones, small shells, sand, and plant materials. There are some that make them in quite haphazard ways, and there are some that make them in very intricate ways. We can identify some of them to species just based on the way they make their case. Then we've got our caseless caddis. They don't live in a case, but they do also use their silk thread to spin a web that they use to strain particles out of the watercourse to feed on. Most of the groups that we look at for this survey are filter feeders, or they feed on algae and detritus. But this one at the top here, the green sedge, the Rhycophilidae, it is a predator, so it actively hunts on the riverbed. So we do find that when we take these out and put them in our tray, sometimes they do start to get a bit aggressive with the other invertebrates in the tray. Then our next group, we've got the upwing flies, known as the Ephemeroptera, and they're named for the short lifespans of their adults. So you might know the mayfly adults don't live for very long. They have no functional mouth parts, so all the energy that they build up is from their larval stage. So river health is really important because the healthier the river, the more energy that the larval stage can build during that phase of its life cycle. So as you can see, all four of these upwing flies that we have, they have three tails. That's really useful for our volunteers to help identify them. So we are looking for the mayfly, the Ephemeridae, the blue wing olive, the Ephemeralidae, the flat-bodied Heptadenidae, also known as a stoneclinger, and an olive, the Betidae, and that's also known as an agile darter. So as you can tell from some of the names, it helps us identify them because of the way they move in the water as well as the way they look. Then we have the stoneflies. These are order Plecoptera, and that's named for the hard, shiny appearance of the wings of the adults. And here you can see that one of their defining features is they have two tails rather than three, and they have these really long antennae. And then finally, we have the freshwater shrimp. So this is the only one of our target groups that spends its whole life cycle in the water. The others, they are the larval stages. So once they emerge as adults, they fly away. But when it comes to the freshwater shrimp, it spends its whole life in the watercourse. So what are the benefits of the survey? So our volunteers are contributing to a UK-wide long-term data set. So as well as observing trends, we can pick up any potential issues with water quality. At Guardians, we've been working with SEPA, and earlier this year, we were able to give all of our groups what is known as a trigger score and also a target score. The trigger score is what is known as the minimal acceptable score for their site. So once they've estimated the abundance of their invertebrates, assigned a score. That score is going to be higher if they have more invertebrates. Then we have what's called a minimal acceptable score. And this sits somewhere on that line between moderate and poor. So we make the assumption that if our score is below that line, that the water quality is poor or bad, and that if it's above that line, it's high, good, or moderate. But as I mentioned, this is an assumption, and it's quite a blunt tool, but it is useful because if we go below that line, there is a procedure to follow. That involves the groups repeating the survey just to make sure that it wasn't an error in their sampling technique. If it's still below that, we can contact SEPA, inform them of the trigger breach, and then there is a response from their local ecologist. So this really empowers our citizen scientists. They're working in and next to the river, getting to know this hidden world of freshwater invertebrates, learning new skills, and providing valuable data. And the groups that have triggered, gone below that trigger score, have been happy with their response from SEPA. They're getting to share knowledge and have conversations with their local SEPA ecologist. So a little case study here. So this is from the River Fly on the Esk group on the Midlothian Esk. So the Esk starts in the Pentlands in the south and then comes out of the mouth in Musselburgh in the north. And this community group was set up in 2019. And 6 years on, they have 17 sites and they have over 50 volunteers trained. And as well as doing the riverfly survey monitoring, each month they also do a simple water testing. So they look at phosphates and nitrates and turbidity. And they've worked with a landowner to plant over 230 trees along a problem bank to stop erosion. So you can see from the graph, the green line is the score over time, and that red line is a minimal acceptable score. And while they haven't gone below that score, they have had two near trigger events. So the local group coordinator could tell that all was not well with their river, and so they reported this to SEPA, and then they found out that there had been a fault with the local water treatment works just upstream of their site. And when they went to do the site survey the following month, then everything had returned back to normal. And then there was another pollution event that happened in Pennycook, and the group were able to send a watch out. So they asked their other riverfly groups downstream of that location to go out and take a sample, and they could pinpoint the trajectory of the pollution. Luckily, it was only detected locally. So this gives you an example of how catchment scale monitoring can help you pinpoint and record the journey of a pollution event. So as we're in the latest phase of this project, we have quite a number of experienced volunteers. And so we've been able to offer them what we call the extended survey training. So this allows individuals to upskill their identification skills because they go from looking for 8 invertebrate taxonomic groups up to 33 taxonomic groups, which sounds like a really big leap, but because they've been doing this for a while, they've already started to notice these extra taxa. It gives them more information about the impact of water quality and as well as siltation and flow. And the training follows a similar pattern. They will have a self-guided theory session. They watch a video for around an hour and do a quiz. And then we come out in person and we do kick sampling. So this is good for them to refresh their memory of how to take a kick sample, but it is slightly different for this survey. We're taking into account surface dwelling invertebrates. So we do a quick sweep, 30-second sweep with our net, just to try and catch any of the surface dwelling invertebrates. Then we go on to look at the taxa that they find in their sample, pointing out those additional taxa that are different from their normal survey. And we also come along with a reference collection to show them the ones that we don't find in their site. So here's just a photo of some of the taxa we look at, some of the invertebrate groups we're looking at in the extended survey. So it still includes the cased caddis and the caseless caddis, the flat-bodieds and the olives, but we've included beetles, worms, leeches, flat-bodieds, dragonflies. So we've included things that are a bit more tolerant to slow flows and siltation. So that gives us an idea of whether the site has been impacted by that. So what are the benefits of doing the extended survey? So the 33 groups does include the majority of invertebrates that we're likely to find in a sample. And it includes, like I said, those groups that are tolerant of organic pollution, low flows and siltation. So that gives us more information about the river's quality and possible stresses to the and its biodiversity. When you enter your data, you get a water quality score and a silt and flow score. So you can keep track of that over time and see how it's changing. And this is particularly important to our groups that are monitoring sites that are near agricultural land, forestry land, or development. And that might be the reason why they've set up that monitoring. So this gives them, they can look at these results and compare it to the land use that's happening around their sites. And it's also really good for groups that are monitoring the impact of river restorations. We have a few groups that are post-river restoration where their water course is constantly changing. There isn't really a normal yet, but it's just really interesting to see how the river is changing and how that's impacting the different invertebrates they're finding over time. And we have other groups that have river restorations planned for the future. So at the moment, they're maybe monitoring quite impacted, heavily modified watercourses. But the idea is that over time, as they start working in the catchment, improving sites, that they'll start to see hopefully an improvement in their riverfly score. Onto the climate change. So, with extreme events such as sustained warm spells and heavy rain becoming more common, um, drier, warmer conditions coupled with increased pressures on water supplies will lead to low flows in headwaters that might stop flowing altogether, leaving behind disconnected pools of water that can become isolated. Headwaters are really important for invertebrates. It's where a lot of the recolonization is happening of invertebrates. They move downstream, so we need the headwaters to be sustainable. An increased water temperature will lead to cold water adapted invertebrates, known as cold stenotherms, to move to higher altitudes and latitudes. And the ability for freshwater invertebrates with a terrestrial adult phase to disperse and find refuge will depend on how far the adults can fly and the length of their flight period. So poorer flyers, such as stonefly adults, are going to find it the hardest to disperse and adapt to climate change. So it's predicted that the higher altitude rivers in Scotland are going to be increasingly important refuge for the upland summer mayfly and the freshwater pearl mussel. The upland summer mayfly lives in harsh habitats in high altitude headwaters that get covered by snow. The snow insulates the water from the lower temperature during the winter. And the snow cover is vital so that the adults can emerge before the water of the headwaters dries out over the summer. But this period is shifting with the rise in air temperature leading to an earlier snow melt. So that emergence of the adult coinciding with the right time is getting smaller and smaller. And it's predicted that the the upland summer mayfly will be restricted to the cairngorms in Scotland as its range of suitable habitat is contracting. And freshwater pearl mussels, they live in clean beds of fast flowing rivers where they can be buried partly or wholly in coarse sand and fine gravel. The mussels have a complex life cycle. In their first year, they live on the gills of young salmon and brown trout, which doesn't cause the fish any harm. Higher temperatures are thought to affect the development of the larval stage of the mussels, and it can disrupt the life cycle timing between the larva and the fish host, which it needs. And the increased occurrence of large flooding events can wash away this gravel and sandy habitat needed for the freshwater pearl mussels, which are also impacted by illegal pearl fishing, poor water quality, and habitat damage. So things that you can do, you can think about your personal carbon emissions, what you eat, what you buy, how you power your home, how you travel, reducing your water uses to keep our rivers and lakes and streams full, planting trees along riverbanks keep for shade and to keep waters cool. Riparian woodland also filters diffuse pollution and reduces soil erosion and contributes to net zero with storing carbon storage and sequestering. Using alternatives to peat in our gardens will keep carbon in the ground and protect essential habitat for thousands of invertebrate species. And restoring wildflower grasslands will help sequester carbon and aid the movement of invertebrates. And here I've just got a screenshot of all the sites that people are monitoring throughout Scotland. I probably should have done a bit of a zoom in so you could see Forth Valley, but we do have a group in Clackmannan. We also have groups in Fife. And if anybody's interested in getting involved with the Riverfly group, please get in contact. Riverfly groups only have one more sample for this year, but we'll be getting people in contact with their local Riverfly groups so that they can get involved with the training, which will begin in spring, and you'll be able to be fully trained and help out the surveys throughout next year. So for the project started back in 2023. We've now trained 58 groups throughout Scotland, and that's over 800 individuals that we've trained. And I'd like to thank all of our volunteers that go out each month to collect the surveys, our funders at National Lottery Heritage Fund, and our partnership with Riverfly Partnership. Thank you very much.
Thank you, Kerry. Wow, that was so good. Thank you. I feel like we got a wee mini training there as well. So much to learn. And so much life in the waters as well, which is always mind-blowing. How much is there. Did anyone want to start off with any questions for Kerry at all before I put her on? Hi, Kelly?
I was just going to ask, when the level of drops below that line and they get in touch with SEPA, what kind of things can SEPA do to kind of combat it or to bring the water quality back up? Or so what do they generally do?
So
I'm
it really
interested.
varies. No, that's okay. It really varies. So there are some groups, so we have a really wide variety of groups and wide variety of locations. We have some groups that are in urban areas that are maybe impacted by mine water. So their response from the SEPA ecologist has been that this is something that they're aware of and that this is a very long-term problem with no quick solution. We've had other SEPA ecologists who've been able to go out and say, actually, I've spoken to the landowner and we've identified a problem. Quite often, when that happens, we don't get a lot of detail about what was discussed because that is between the landowner and SEPA. We have had other incidences where we think the trigger score has been breached due to works happening on the bank, so increased siltation. And again, the SEPA ecologist was able to inform the relevant person within SEPA that the work was going on, and they had a word with the landowner. So it really varies. A lot of these things are long term. Not very many of them are quick fixes. Quite often, we find that by the time the SEPA ecologist is able to go out, um, the pollution, if there was a pollution event, it might have washed away. But it's all still really useful information. Um, so yeah, it really varies. But yeah, so far it's been quite nice. I think that a lot of the groups that have uh, reached their trigger levels have had a conversation that they wouldn't have had otherwise, because they've been able to have that contact with their SEPA ecologist. But something I forgot to mention in the talk actually was that with the low flows and high temperatures this summer, that has kind of exacerbated some of these trigger scores. So we have given our groups advice about when it's too hot to sample, when the water levels are too low to sample, and just giving people a bit of a heads up and say, you know, if you go to your site, it looks pristine, but you get a low score, then that might be because of the high temperatures. So in those instances, we've had a conversation and maybe decided not to pass that information on to SEPA, and other times we have. So yeah, it's been interesting, just the added complexity of this really hot, dry, long period that we've had.
Thank you. Cheers.
Thank you. Yeah, I think a lot of our communities are kind of doing resilience planning and looking at flooding and things. So it's almost like quite a nice tie in that longer term sort of awareness of your area and how well every part of it is doing.
Yeah, it's a really good
Right.
tool. Like if you have the plan to, raise money to do restoration projects or flood resilience projects, uh, getting a Riverfly survey set up
Hmm.
early and you can use that data. You can see that data change as the project moves on. So yeah, it's a really good tool to use. We've got some groups that are using Riverfly um, as their tool to gain more funding. So yeah, it's quite a useful way to monitor, but also to sort of make your group sustainable and find out data that's useful to help you in other ways.
That's really cool. So interesting. I was quite keen to know, at the start when you're starting up groups, just bearing, in mind Forth Valley and things and how we could maybe support. How do you, what are your tips for sort of establishing that engagement, maybe with folks who don't see themselves as scientists, but how do they get involved? How do you sort of entice them in to becoming involved?
I've been really lucky that I joined the project once it was about a year old, and I haven't had to do much convincing people. It's really spoken for itself. People are really enthusiastic. Um, So generally we have community groups approaching us or individuals approaching us on how to set up a community group. We help with that too. But conversely, we do sometimes have groups that would like to maybe do something like this, but they're not sure if it's for them. They're not sure about the commitment. And so I do deliver talks like this. So sometimes I can deliver talk to a group that already exists, talk them through similar what we did today, let them know about what's expected of them, what the commitment is. And then sometimes that breaks down the barrier and then people are much more likely to get involved. And then if maybe an individual is struggling to get a group of people locally, maybe I can connect them with another group where they can join. Quite often, people who are involved in a group can then kind of splinter off and create their own group because they have the experience. But sometimes it can be quite difficult if you're an individual in a community and you're struggling to get other people to get involved. That can we have had a few examples where that has taken a bit of time to get them to a level where they have a group of people who can get trained. But I'd say majority of the time, a lot of people have come to us as a group, or we've just had luckily a number of inquiries from a similar area and we're able to put them all together to form a group.
Oh, great. Thank you. And Kelly, did you have another question?
Well, it's kind of a follow on from what you were saying earlier. I just to sort of clarify a little bit because quite a few of the groups I've been working with are quite interested in adaptation planning and resilience planning. And that's something that certainly in the last few months, last year, that's really starting to come online now. And I suppose back to what you were saying before. So do you think having doing something like the Riverfly survey would be quite a good thing for community groups to do to kind of create that data for adaptation planning and the sort of discussing around those sort of areas? I'm just sort of thinking about 'cause one thing that does come up is talking about biodiversity and climate change and how to sort of make their communities more resilient in relation to that. So being able to have that data, do you think it would be kind of a useful thing for communities to do
Yeah,
in that
definitely. I
sense?
mean, when it comes to monitoring, you need to know what's there to be able to know
Mm,
what you're protecting or what you want to protect. You need to know if it's in decline or if it's increasing, how it's been impacted. So
mm,
monitoring and having data is never, just for the sake of it. It can be applied to so many things.
yeah.
So yeah, I definitely recommend it to groups we have quite a few groups, climate action groups, sustainability groups that are doing lots of other things. And Riverfly really fits into their sort of ethos and their other activities that they're doing as a group.
Mm, good, thank you.
Thank you. Any more questions? I think you shared some really helpful resources there as well, Kerry. Thank you. So we'll put that up on our blog following this too, about how people can get involved and join forces because it sounds like a really wonderful project. I'm sure you also get sort of lovely feedback of people just enjoying being in nature and
We do actually. I did actually have a feedback slide and I removed it. I should have kept it because one of my best ever feedback was an angler who was looking at one of the mayflies and he said, "This is the second best day of my life."
oh,
And it's always stayed with me. But we do get really nice feedback. People are really enthusiastic, and it always blows me away how much people are happy to do the training. It's 6 hours is quite a lot of time to devote to training, and then each month they're going out and surveying. That's roughly 2 hours each time they go out. So yeah, it just it blows me away people's enthusiasm and their commitment. Obviously I'm biased. This is something I do for a living and something I've been really passionate about for a long time, but it's really nice that we can infuse other people and pass on our knowledge. So.
yeah, absolutely. And I think, when you're in a group, they're probably enjoying all the social connective.
Yeah, yes,
There's lots of
definitely.
stuff.
A lot of groups, they like to bring cake along and have a nice chat. And, especially, I mean, some of these lovely sunny days we had this summer, it is sometimes, it feels just lovely to be on the riverside chatting and showing people invertebrates. And yeah, great.
No, yeah, it sounds super lovely. I'm very keen to get involved after.
Brilliant. Yeah, so I could send you the link to our webpage on the Buglife website, and we could maybe add that, and people can get in touch through that way to let me know if
Yeah,
they're interested in Guardians.
Absolutely. Yeah. So, thank you so much for sharing that today. And sorry that you're the solo speaker, all the
Oh, that's
emphasis
okay.
on you today, but it's been really, really helpful and nice that we could relax and just hear a lot of information there as well. So thanks for the dual purpose. I feel like it was a bit of training and also just sharing about the project. So really appreciate your time. And yeah, thank you everyone for listening in and taking part. Links into the chat to the FEL's website where we can share things like the blog and that kind of thing too. Um, so thank you. Yeah, I'll let you all get on and enjoy the rest of your lunchtime.